Hi, everyone, and welcome to our last video lecture for the semester. Congratulations. So in today's lab, or this week's lab, I should say, we're going to be focusing on our special senses, primarily the ear and the eye. So let's get started. Now we're going to start first with the eye. The eye has two sort of categories of structures, those that help with actual process of vision. So those are going to be like the main structures. And then we have accessory structures like those you see here. So the ones that we're going to be focused on in lab are the structures that are going to help move your eye. So the extrinsic eye muscles, as well as those that are going to help with protection. So that includes a group of structures called the lacrimal apparatus. We'll talk more about some of these other components in lecture. So with our extrinsic eye muscles, they're pretty straightforward in terms of recognition. So if we think about some of our old terminology, rectus means straight. So the rectus muscles are the ones that are going to be here forming sort of a cross. So top, bottom, out, and in. And then we have two oblique muscles. So they're going to kind of run on the top and bottom of the eye like this. In fact, they're important for rolling the eyeball. So we have an inferior rectus and a superior rectus. So in this unit, not only are we identifying them, but be prepared for me to ask you, what movement does this eye muscle do? All correlated to this particular diagram. Now, I want you to remember this particular muscle here, the superior oblique. It's very helpful for determining medial and lateral if we get an eyeball just sitting out like this. The superior rectus, if you notice, usually a tendon is how we see it on the models. The tendon always points to midline. So this muscle always points to the midline of the face. So that lets you know this is medial and this is lateral. So that's one of two ways that we'll talk about recognizing those directional areas on the eye. Now, the lacrimal apparatus has a job of protection. There's lots of things that we have, our mucous membranes, our eyelids, our eyelashes are all there to help protect the eye. And one of the other components we have are the tears, the fluid that's around the eye and drains from the eye. And it's produced and drained through the structures of the lacrimal apparatus. So three that we're going to be observing in lab are the lacrimal gland, the lacrimal sac, and the nasolacrimal duct. So the lacrimal gland, this is our second way to determine medial and lateral. The lacrimal gland is always on the lateral side of the eyeball. So the tears are formed in the lacrimal gland. They go onto the eyeball, we blink, and that pushes it towards midline. They're going to drain through structures that we'll talk more about in lecture, ultimately ending here in what's called the lacrimal sac. From there, we drain down into the nasolacrimal duct. And this should look familiar. We have our eye in orbit model that we'll be using this week that showcases these structures. So now it's time for the eyeball. When you look at an eyeball, it has three major layers to it. A very tough outer layer called the fibrous layer or fibrous tunic, an inner layer where most of the structures are called the vascular tunic or vascular layer, and then the deepest layer at the back, which is the retina or neural layer. And this is where we get the exchange of light into electricity that allows us to view images. So we're going to take it layer by layer. So if we look at the fibrous tunic, the two really, really important structures we find here are the sclera, which is the white part of the eye, and the cornea, which is this anterior outward bending of the eye. So the sclera, again, is white. It's the protective part of the eye that's going to give shape to the eyeball. The cornea is clear to allow light to come in and then get transduction down into a nervous impulse. The cornea has a bend to it, and it's important along with a few other structures that we'll talk about later in terms of bending light, so it hits just right on the back of the retina. So the curvature here is super, super important. And we'll talk more about this in lecture, and you'll go into it more detail in physiology. Now, the middle layer, as I mentioned, this is where most of the structures we're going to be learning are located. So the vascular tunic is made up of a dark layer. It's usually represented as red, though when we look at it in real life, it's actually black. This is called the choroid. This is the posterior part here. It's a very dark layer, helps collect excess light rays. On the anterior portion, we're going to see these sort of bulbous areas that are attached to strings. This is called the ciliary body. Within the ciliary body, we have ciliary muscles. On most of our models, we don't distinguish them, so I'm okay with you using either term to identify. The ciliary bodies are attached to this structure here called the lens through these suspensory ligaments. And their job is to stretch and squish the lens. Again, this is one of the structures that helps with focusing the light. The ciliary body is also an important region because it produces one of the two fluids of the eye. The fluid that circulates in the anterior portion of the eye is called the aqueous humor. And it's, as its name dictates, very liquidy. So it's produced here in the ciliary body, and it circulates through the anterior portion of the eye, and then is reabsorbed through these areas here called the scleral venous sinus. Now, anterior to the lens is the colored portion of the eye called the iris. It's made up of two different types of smooth muscle. Again, it's going to change shape, it's muscle. And when it changes shape, it changes the shape of the pupil, which is the black spot of our eye, which is the opening into the eye. And we'll talk more about how it changes and why it changes when we get into lecture. But the iris will change shape, allowing more or less light into the eye. And the pupil, again, is where that light is coming into, the window to the eye. Now, in terms of the eyeball, we have cavities and we have chambers. So in the lab, we're primarily focused with segments or cavities. So the anterior segment or cavity is everything in front of the lens. So this includes the ciliary body, the lens, the aqueous humor, the cornea, the pupil, and the iris. Everything behind the lens is filled with the second fluid, which is called the vitreous humor or vitreous body. And so the entire posterior cavity is filled with this particular fluid. Now, it's more of a viscous fluid, as you guys will see when we get to the eye dissection, but it helps keep the retina up against the back of the eyeball, again, allowing focus to occur. And so we have two distinct fluids, aqueous humor in the front, allowing nourishment of the eye, helping with bending of the light, vitreous body, maintaining the retina against the back of the eye, as well as providing bending or refraction. Now, the retina itself, we'll take a look at it in a minute, is divided into two layers. So we have the pigmented layer, which is on the back, and it's again going to absorb light just like the choroid does. We want as much light to be pinpoint focused on that area of high vision than have a lot of it scattered. It makes sure, that's what glare is essentially. And then anterior to the pigmented layer, we have what's called the neural layer. This is where all of the nervous tissues are. So I'm just going to kind of talk about these structures and then we'll look at them on the next slide. So we have a layer of photoreceptors. These are your rods and cones. They're the ones that are responding to changes in light. So lots of light or not a lot of light. When they get excited, they're going to send their signals anterior to the bipolar cells and then further anterior to the ganglion cells. And the axons of the ganglion cells are all going to converge on a single point of the eye. This is called the optic disc, and it is where your optic nerve forms and exits the eyeball. This area is known as your blind spot because we have no rods and cones, so therefore you don't see anything. So this is what we're talking about. On the left is a figure, and on the right is the actual histology of the retina, which we'll be observing in class. As you can see here, we have the pigmented layer. It's going to absorb extra light. We have our, excuse me, our nuclei of our rods and cones, then the bipolar cells and the ganglion cells. Now, right now for us, it's not important how the signal is moving or what goes on at each of these layers. That's a physiology concern. But to help you guys in understanding where things are, you want to think in terms of number. So the rods and cones, there's a lot of them. So that layer is going to have a lot of nuclei. There are fewer bipolar cells and even fewer ganglion cells. So on a quiz, on an exam, if you're looking and you see a layer with a lot of nuclei, those are the rods and cones, fewer bipolar cells, even fewer ganglion cells. So this is one of the two histologies we'll be viewing this week. Now the retina, when you look at it, as you do here, this is what your optometrist sees. We have our optic disc, and you can recognize it because we have the branches of the retinal artery coming into the eyeball. This is where the nerve is leaving. So again, you can't see anything here because there's no receptors. If you move laterally, you'll see sort of a dark smudge here. This is called the macula or the macula lutea, and this is where the light is focusing on the retina. It is sort of our HD vision. Now, in the center of the macula is a depression called the fovea centralis. It literally means the central indentation. And this is where we only have cones. So these are the photoreceptors that are responsible for detecting color vision. So this is your area of greatest sort of sharpest image. How do we tell the difference on a model? On an exam, if I ask you just to identify the area, it's the macula. If I say identify the indentation at the center, that would indicate I'm looking for the fovea centralis. We do have one model that actually depicts the actual indentation, so please make sure you take a look at that one to really understand the structure. Now for our final dissection of the year, we're going to be cutting open sheep eyes. And the protocol is all in your lab packet, so I'm not going to worry about that now. Basically, you're going to cut the eyeball into anterior and posterior sections, and you're going to be working to identify all of these structures. So I have some references here, but the one thing I do want to talk about is a new structure called the tapetum lucidum. So this is a modified choroid, only seen in animals that can sort of see at night. It's a very shiny, reflective blue-green color, and it lets the light bounce around in the eyeball to activate those photoreceptors. So when you shine a light in your dog or cat's eye and it reflects back at you, that's what you're seeing, okay? Now, we can also see the optic disc here. The easiest way to find it is see where all of the wrinkles of the retina converge here in one singular spot. That is your blind spot. And so that's one of the easier ones to see on the sheep eye. So before we move on to the ear, I just want to say again, I haven't gone through every single structure we're looking at, but that is a good overview of the eye that you should be able to master all the structures on your list this week in lab. So if we move on to the ear, which is relatively simpler, we will notice some similarities. We have three regions: an outer, a middle, and an inner ear. And the deepest layer is where we have our receptors to protect them from the environment. Now, the outer ear's job is to get the sound into the ear. So your outer ear, which we colloquially call the ear, is actually called the auricle or the pinna. We have the helix, which is that upper lobe or upper sort of bend of cartilage on your ear. And then you have down here at the bottom, that's your lobule. So these are different curvatures of cartilage to help gather sound waves and direct them into your ear canal, which for us, because we're in anatomy, it's called the external acoustic or auditory meatus. We learned about it earlier in our bone lab. So the sound waves are going to funnel through here and hit this membrane. This is your eardrum. Again, we're in anatomy, so it is the tympanic membrane. This is the point where we go from a wave to a vibration. And that's actually what your receptors in your inner ear detect. They don't detect sound waves, they detect vibrations and movements due to those vibrations. Now, the eardrum, the tympanic membrane, is the entry point into the middle ear. The middle ear has our three bones, which we call the ossicles, the malleus, the incus, and the stapes, or the stapes. Now, these you are going to need to recognize individually. The stapes more often than not, is going to be physically attached to the inner ear over an opening that we call the oval window. So what happens is the vibrations from the tympanic membrane vibrate the ossicles. And as the stapes pushes into the oval window, it's going to cause a movement of fluid within the inner ear. So again, we don't actually get to see the oval window. So if I wanted to ask that, I would just point here and say, what is this opening that lies under here? We also have another opening in the middle ear right here called the round window. As this cochlea here, this is where our sound receptors are. It is filled with fluid. You cannot compress a fluid. If you try to compress a fluid, it's going to want to escape. So what we have is the sort of second tympanic membrane. As the stapes pushes in through vibrations into the oval window, the membrane over the round window bulges out, giving the fluid somewhere to move. So it's almost like a piston. Push in, push out, push in, push out. And so that's how we're able to keep those fluid waves moving through the cochlea. Now, the final structure I want to show you is the eustachian tube, or the pharyngeotympanic tube. This is a tube that runs from the middle ear down to the pharynx, and it helps equilibrate pressure. So when you're popping your ears, you're basically opening the eustachian tube and allowing a transition of pressure to occur. In kids, it's very short, which is why they tend to get ear infections more often than adults do. So they'll have a cold, it travels up the short eustachian tube, and now we have an ear infection. Now, the inner ear is where all the receptors are, and the ear is a very special organ in the fact that it's responsible for two separate special senses. Hearing, which is all done here in the cochlea, and all those structures we've talked about lead to that end result. And then we have balance, or equilibrium, which is all detected up here. So this portion here contains two areas. We have a big fat area, which is the vestibule, and then we have our canals here, the semicircular canals. Both of them are detecting acceleration and deceleration, so movement and stopping of your body and head. And we'll go into a little bit more detail and lecture about exactly what you're experiencing. And of course, you'll hit it very hard in physiology. So for us, each of these semicircular canals are oriented in a different plane to monitor different movements. So we have an anterior, which comes at you, lateral lays on its side, and the posterior canal is the one that's in the back. So anterior comes at you, lateral lays on its side, and the posterior is the one at the back. Okay, so even though these are two separate senses, they are all going to share a common route to the brain, which is through cranial nerve 8, the vestibulocochlear nerve. And again, more information on that pathway in lecture. So the cochlea is where hearing is interpreted. It is made of a spiral that has 3 separate chambers in it. And when we cut down the middle of the cochlea, this is what we see. We have a top chamber filled with fluid called the scala vestibuli, a bottom chamber, also fluid filled, called the scala tympani, and then in the middle, we have our cochlear duct. This is what's found in the fluid-filled cochlear duct, the organ of Corti. This is what is detecting and sending signals to the brain about sound. So as the fluid moves from the vibrations of the stapes, it's going to cause this membrane here called the basilar membrane to undulate. And these little hair cells are attached to nerve fibers, are going to push up against this rigid membrane called the tectorial membrane, kind of like this. And depending on how they move, it's going to send more or less signals to the brain, and that will be part of a pathway that lets you know what you're hearing, what it sounds like, and where it's coming from. Now, these chambers are divided. We already talked about the basilar membrane on the bottom. That separates the cochlear duct from the scala tympani. And on the top, we have a very thin membrane called the vestibular membrane that separates the scala vestibuli from the cochlear duct. Now, when you look at the section of the cochlea, we have a bony area running through the middle here called the modiolus. It's basically the axis around which the spiral forms. So think of like icing a cupcake or making an ice cream cone. You follow sort of an invisible axis. In this case, it's not so invisible, it's bone. And so the chambers that we just talked about swirl around this modiolus. These chambers are completely separate until we hit the apex of the cochlea, where you'll see, if you look closely, they merge together. This merging, and you'll also see an accompanying depression on the outside, is called the helicotrema. It's where they meet together so that it can bounce back outwards. So you kind of think of sound waves moving all the way to the top through one chamber, and they bounce back and come back down through another chamber. It's kind of a cool way of thinking about it. So here is an actual histological image of the cochlea. It's very similar to what you guys will see in lab. Granted, it's a perfect specimen. What you guys are more likely to see, though, is we're going to zoom in only on one of these sets of chambers. And what we typically see is the vestibular membrane here is often broken because it is so delicate and very thin. And we tend to see the tectorial membrane, instead of being nice and like laying on top of the hair cells here, like we can see in this diagram, it likes to flip backwards. So just be aware, it's not going to be as perfect as what you see on this image, but we can see all three chambers, we can see the membranes, we can look and see the modiolus. It's really neat. So this is our final histology of the year. So what are we looking at? Well, I have lots and lots of eye models, a couple that are standing, one's big, one's small. We have our eye in orbit. I have another eye that has a lacrimal gland on it, and I have a couple smaller eyes floating around that are a little bit older. I have two ears that look exactly the same, and I have one that's again a little bit older, but it's still very good in showing a lot of the structures. And the final model we have is this one here, which is of the organ of Corti. So we can see a lot of the structures that we just talked about. So guys, that is it. This is our final lab and our final lab lecture, and I will see you there. Bye.